All right, good afternoon. If everybody could grab a seat, we'll get the afternoon session kicked off. So I'm Michael Martin. I'm a AAAS Science and Technology Policy Fellow at Oscar. If you want to know what a Science and Technology Policy Fellow is, hit me up afterwards. And our first speaker is Heather Mays, who recently completed her PhD at Northwestern in chemical engineering. Her advisor is Linda Broadbelt. She did her practicum at NREL, and she has already accepted a postdoctoral position at the University of Chicago, and then will join the faculty at the University of Michigan. So.
Thank you very much. Thank you all for coming to my session right after lunch, and I'm very excited to share with you some of the work that the CSGF funding has enabled me to do. My motivation for the work is to move from a non-renewable energy source, obviously, like to get here on a plane, started by taking carbon that was sequestered in the ground already, converting it to a fuel, releasing it to useful energy that got me here today, for which I'm very glad, release CO2, but obviously more and more CO2 will be released. My research focuses on going towards a renewable cycle. And you may remember a few years back, the fellows that were here at the conference, Stephen Chu came. He was still the US Secretary of Energy. And one of the things I found inspiring about him is that his background isn't energy, yet he ended up in this position. His Nobel Prize was in physics. And when I asked him what made him switch from physics, where he got his Nobel Prize for optical trapping, to worrying about problems of renewable energies, he believes this is the existential crisis of our day. So I appreciate the funding to help us work on this problem. So one of the ways that we can use a renewable source of energy to fly me on a plane here is if we can take biomass, waste biomass, and convert it into a liquid bio-oil that could be upgraded into a fuel, like a jet fuel. And one of the technologies that's very promising for this is called fast pyrolysis. This process, we know how to do it. I could make you bio-oil today if I had the right equipment in front of me. But I wouldn't be able to tell you much about that bio-oil. I wouldn't be able to tell you what molecules are in it, because the composition of that bio-oil depends greatly on the composition of the biomass, which changes depending on the plant, depending on the region, as well as on the conditions in the reactor. But of course, we have very specific specifications for liquid fuels. So what we'd love to have is a computational model that could describe the transformations that happen in fast pyrolysis. The first types of models that were available, a very famous model in the fast pyrolysis community is called the Broido-Shafizadeh model. And this model is very simply solved mathematically, obviously. It's a great virtue. You need very little data. These are basically lumped components, and this is just saying, let me just look at the largest component of biomass, which is cellulose. And I'm going to hypothesize that I can represent all the reactions that happen in just a few steps and use some experimental data to fit these parameters. The limit is that you can't get a lot of data about exactly like what these volatiles, this would be the liquid oil, what components those are. You can still use an empirical model to have a little bit more complexity, but again, this is going to suffer from needing some experimental data to fit these parameters, and then the parameters will only be valid within the range of data you used. Also, if I wanted to add in more components than just cellulose into this model, I'd have to throw the model away, start with a new, start with a new setup, a new hypothesis, and have even more experimental data to fit this. And again, it would not be valid beyond the data which was used to fit this model. So how else could we model biomass fast pyrolysis? The Broadbelt Lab, we specialize in microkinetic modeling. And the fundamental differences between a microkinetic model and an empirical model is we start from the basic chemistry. We start with the actual species, the actual components that are the reactants, the products, and the intermediates. And then we connect those by understanding the reaction network. What is the chemistry that can convert one molecule to another? So I hope you all see that this is very much, it's just solving a graph problem where the thermodynamics determines property about the nodes, and the kinetics determines basically the weights of the edges, how fast you move from one to the other. And one of the great things about microkinetic modeling is because it's based on the actual chemistry, it's fully extendable. If I learn that there's a reaction path that I didn't know about when I first built the model, I can simply add it in. Everything I had before was still valid. Likewise, if I learn by a new experimental technique that there was a product I didn't originally capture, you can add that in. And then say if I start with a model of cellulose pyrolysis and then I want to start adding in more components of real biomass, again, I can add that to my network, figure out how it fits into the model. So it's completely extendable. And because it's based on actual chemistry, it's valid at any conditions. I don't need to do a lot of training. You have a lot of training data to fit empirical parameters. So why was there not a microkinetic model of even just pure cellulose fast pyrolysis when I started my funding by CSGF? Well, because we'd have to know all the species, including the intermediates, and all the reaction paths. Biomass fast pyrolysis happens at about 500 degrees C on the order of seconds, and many different reactions are enabled at that temperature, and experimentally it's not feasible to separate them apart and look at them, but computationally there is. So that's what we focused on, is how do I computationally determine properties of these nodes and edges. And one of the most important edges in this graph of biomass fast pyrolysis is this glycosidic bond cleavage. This reaction pathway accounts for 59% weight of the product yield. And yet we did not know the mechanism of this, and therefore we did not know the kinetics, not for lack of trying. This reaction had actually been studied for over 90 years, and it was still debated whether this happens through a homolytic, a radical mechanism, or a heterolytic, an ionic mechanism. This was actually studied long before looking for biomass because cotton is almost 100% cellulose. So people have been looking at these reactions for years, even just for flame retardants for clothing. And yet it could not be determined experimentally which of these were more likely. But computationally, using DFT, using quantum mechanics, we can explore what reactions are possible and what the energetics and kinetics would be, and therefore what would be most likely. One of the big computational challenges of this work is not just the number of atoms, but the conformational flexibility. These rings are extremely flexible. These exocyclic arms are extremely flexible, and so there's a large potential energy surface space that you have to explore in order to find the most relevant properties. And in the process of investigating which of those two theories was most likely, I found a different possible mechanism, which I call the concerted mechanism. In the other two, you got very reactive intermediates. In this mechanism, we simultaneously form and break bonds in single steps. And then we could use our results for DFT to see which one is more likely. And to give you an idea of what kind of the scale means here, every 3.5 kcal per mole difference at 500 degrees C corresponds to about an order of magnitude difference in the rate. So it looks like in the concerted mechanism that I discovered is most likely. But this slide alone cannot tell us if any of these three mechanisms can happen. What we do is we put it back into the microkinetic model and then solve that model in time and see if we can come up with predictions at different temperatures for this levoglucosan yield. And using this new paradigm that we came up with, we're able to match experimental results at a variety of temperatures, giving us confidence in the mechanism that we have the time dependence correct. the temperature dependence correctly. While I was working on all this thermochemical transformation of biomass, I then went off and I did my practicum at the National Renewable Energy Lab, where people were looking at how organisms break down these bonds. And what's very interesting about the glycosidic bond is these are really strong bonds. This is why your cotton shirt you can wear over and over again, throw it in the wash, it doesn't fall apart. The estimated half-life, if you just left cellulose glycosidic bonds by themselves, is over 4 million years. And we've actually found chemically intact cellulose even older than that. So why is it that when I go walking through a forest and all this biomass, I'm not walking through feet and feet and feet of it? And it's because organisms like fungi make enzymes that can break down these very strong bonds at ambient temperatures. So that got me interested in looking at these mechanisms. And the importance of understanding how the mechanisms of these cellulases is because they're actually currently in use industrially to break down biomass into these first cellulosic ethanol plants that opened last year. And experimentalists who are working on trying to breed more and more efficient enzymes have found that understanding the mechanism directs them to have their experiments targeted to find more efficient enzymes more quickly. So we wanted to use computation to look at the actual details. Experimentally, crystallographers can get pictures of enzymes. This is, for example, discovered in 1990, the Trichodoma ricei cell 6A, the first cellulase that was crystallized. And experimentally, we can get these static pictures. But computationally, we can actually start to explore what are the interactions between different residues in a protein that make this reaction happen at low temperature, and understand why some features are consistently observed. For example, this bond shown here, this is this really strong glycosidic bond, and over many different cellulases working on many different sugars, it's observed that right next to where that bond is broken, that subunit is always perturbed from a chair conformation, which you may remember if you took some basic chemistry many years ago, to a pucker geometry. And this is a consistent feature, and the kind of puckering depends on what the substrate is. So one thing that we could do computationally that we couldn't do experimentally is actually make models of all the different ways that these rings pucker. There's actually 38 different puckering geometries that the chemistry body, the IUPAC, recognizes. And back actually in the 70s, Kramer and Popel came up with a way to basically there's two ways for a chair, an envelope, boat, there's a half chair, and a skew. And back in the 1970s, Kramer and Popel came up with a mathematical transformation, taking the coordinates of these atoms in the ring and transforming them into this spherical these spherical coordinates that can exactly describe all the geometries. So using this kind of algorithm, we were able to computationally create all these different kinds of puckers and then use our quantum mechanics to understand what are the properties that are so different that the enzymes find it so useful to pucker them. So I won't go into all the details about them, but this is where HPC was very necessary because there were so many different ways to pucker, because each of these exocyclic groups, you also have the freedom to rotate. And we did a study for 5 different sugars and were able to analyze the differences to help us understand what makes these enzymes so effective. We also wanted to look at other parts of the mechanism. So this T ricei cell 6A I showed you, here's a cartoon, a simplified cartoon of the mechanism that we postulated. But even this, though it's simplified, is back actually from a chemistry standpoint complicated, because you have 4 bonds breaking and 4 bonds forming, postulated in a simultaneous fashion. This is the kind of postulate that's very hard to test experimentally, especially because there are water molecules involved. And with experimentally, you might be able to change the residues, but not the water. And so we can use algorithms, again, kind of tying in how my work really does use the HPC, the algorithms, and the scientific background. And we used a particular algorithm that allowed us to efficiently sample the transition states. These are rare events in molecular dynamics simulations without biasing the potential. But the result of that was that we were able to actually model that this, and I'm just orienting you towards between the 3D view and the 2D view. The green is the glycosidic bond. The H is the acidic proton that's going to help break that glycosidic bond. And we can see if we can see this reaction happen. Here's the nucleophilic water that should go and attack the, this is our puckered substrate. Here's a bridge water that can help transfer the proton to D175. So now, from our molecular dynamics simulations, we can get this kind of data. We can test this hypothesis, and you can watch the reactions happen. Again, something that we can add to our experimental understanding. So where we were, to sum up, where we were when I started my funding by the CSGF is that we had identified a process to make liquid bio-oil from renewable fuels. But we had limited types of mathematical models and questions about the actual chemistry. And in terms of the biology, we had clues that there were some parts of how the enzymes works that were important, but we didn't know why. And we had postulates of mechanisms that we were not able to test experimentally. But using the combination of the HPC, the algorithms, and our scientific understanding, we were able to come up with basic understanding that fed in models. Didn't go into it, but we also looked at the effect of ions on these reactions, as well as understanding what's so special about puckering and understanding more about this industrially important enzyme. With that, I would like to thank, starting with my advisors, Linda Broadbelt, who is wonderful and will actually be here on Thursday morning if you want to meet her. Greg Beckham, who I picked up as a co-advisor at NREL during my time there, and George Schatz, who's a very helpful member of my thesis committee. I'd like to thank all my collaborators and funding, especially the DOE CSGF and the Krell staff who have been wonderful the whole time through. I want to thank various computational resources for my work, and with that, ask for any questions. Thank you very much.
I think we have time